Assalamu alaikum everyone. Welcome back to my channel. So today we'll discuss about the human genome. That is basically the human genome categorization. How human genome is categorized or classified into the major divisions. So however, a human genome is basically the 3.2 billion base pairs long. This human genome here you can see, let me select this. This human genome is basically the 3.2 billion base pair longs that is packaged into the 23 pairs of the chromosomes. So this is classified into the two major divisions. These two major divisions are referred to as the protein coding genome and a non-coding genome. Here, if you look at this pie chart, according to this, the majority of the human genome, that is greater than 98% of the human genome, is non-coding. So you don't think of it as a useless, because it plays a vital role in the structure and regulation as well as evolution. So what does that mean is this 1.5% of the genome, that is referred to as a protein coding genome. Protein coding genome includes the genes that are transcribed and then translated into the proteins. And there are more than 20,000 in just that 1.5% of the genome. So let me write that this small portion of the genome, that is containing the more than 20,000 of the genes that are present in this portion of the genome. So it basically consists of the genes that are first they are transcribed and then translated into the proteins. And within these genes, there are specific regions that are the protein coding regions that are ultimately turning into the proteins. These are the exons. So these major portions that are included in the protein coding genome. So first of all, we have a DNA. DNA is basically transcribed into the useful type of the RNA, that is the messenger RNA. So this is the only type of the RNA that has to be translated into the proteins. So these proteins are then have their uh, 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 vital importance. They have importance in their structural, functional, and they have their regulatory importance. So this transcription is basically a process in a biology in which this double-stranded DNA is transcribed into this single-stranded RNA with the help of a particular enzyme, that is the RNA polymerase. But this RNA polymerase will only transcribe those regions that are meant for turning into the protein. So these specific regions that are you can say these are the coding regions of the genes that have specific instructions of the gene, specific instructions to turn into the proteins. So this coding region of the DNA, this protein coding region of the genome is basically consists of the genes that are transcribed and translated into the proteins, as well as these specific regions, these in green. These are the exons that are the coding regions that are found within the genes. So if we look at the this major division of a non-coding genome, that is approximately 98.5% of the genome is the non-coding genome. Hmm. So this non-coding genome is basically not translated into the proteins. So it has vital roles in the regulation and the various structural roles. So this non-coding region of the genome is divided into three categories. These are the intragenic sequences, intergenic sequences, and the repetitive sequences. Although these intragenic and intergenic sequences, they both comprised of approximately 50% of the total non-coding genome. So these intragenic and intergenic, these are basically the non-coding sequences that codes for nothing, but these have their own importance. They can act as the promoters, they can act as the UTRs, these can be enhancers. So what does that mean is, first of all, let's talk about the intragenic sequences. These intra means within. So these intragenic sequences, these are the specific sequences here. So these sequences are the non-coding sequences. These are the non-coding sequences that are present within that particular gene, that are present within particular gene. So these are referred to as the intragenic sequences. These includes the introns and exons. For example, if this is the particular gene, so this gene is containing the introns and exons. So these gene basically containing the coding versus and non-coding regions. So these, as I already told you, that these regions in the green, these are the exons, and these regions in the blue, these are the introns. These exons codes for the specific proteins, but these regions that are the introns, that are basically spliced out of the messenger RNA before the process of the translation. So these regions that are present within the genes, these do not code for anything. So these will not be translated into the proteins. So these are the introns that are categorized as the intragenic non-coding regions, intragenic non-coding sequences that are present within the within gene. So these are the introns, and other one are the UTRs. These are also present within the genes. As you know that these exons and introns, these are present between the start and stop codons. As you know, at the start of every gene, there is a region that is known as the start codon. And then at the end of the gene, that is the stop codon. That is, I'm talking about the translated regions. So these UTRs are these specialized untranslated regions that are present outside the start and stop codons. Because these regions are not meant for translation. These are not meant for be turning into the proteins. So these are the specific untranslated regions that are present outside the start and stop codons. Next, we talk about the intergenic regions. These intergenic regions, as it clear that this inter means within, this inter means between. So when we talk about the intergenic region, so this intergenic means it is, these are the non-coding sequences. These are the non-coding sequences that are present between the genes, that that are present between the genes. That is, there are many genes. So between these genes, there are specified non-coding sequences that are present. These are referred to as the intergenic sequences. They have their own importance. For example, these includes the enhancer sequences. These are the promoter sequences. These can be silencer sequences. For example, these special sequences, promoter regions जो होते हैं, these are basically useful for the process of the transcription. These are helpful for initiating the process of transcription. As I already told you that the transcription is the process uh, in which the process of basically the copying this DNA into the single-stranded RNA. So this is done by this player, that is the RNA polymerase. It will copy the instruction, copy the information that is found in the DNA and convert it into the RNA. So this information that was stored in the DNA now converted into the RNA. So this is basically done with the help of the RNA polymerase. But this RNA polymerase basically wants to transcribe the regions, specific regions that are called genes. These genes basically provide the correct instructions for making the useful RNA. Now the question is basically, How does this RNA polymerase knows where to transcribe? Okay, how this RNA polymerase basically knows okay, where to transcribe. Now here comes the promoters. Here comes the role of promoters. So this RNA polymerase make use of promoters. So these are the specialized, specific, non-coding DNA sequences that are present in the beginning of the genes. So these provide, you can say, a landing pad for the RNA polymerase and other proteins that, all together will initiate the process of the transcription. So this promoter basically helps the gene to get activated, helps the gene in its activation. So these are basically referred to as the intergenic sequences that are present between the genes. These promoters are helpful for the initiation of the process of the transcription. Now, these are the intergenic sequences. When it comes to the repetitive sequences, these repetitive sequences, a major chunk of the sequences that is greater than the 50% of the sequences, 50% of the genome in the non-coding uh, genome refers to as the repeat sequences or DNA repeats, or you can say as these are the repetitive sequences. These includes microsatellite, mini satellites, transposable elements. These are lines, signs, LTRs that we'll discuss inshallah in our next lecture. Now, if I have to summarize this lecture, so this is the human genomes categorization. Human genome is simply classified into the two major divisions. One is the coding genome and other one is the non-coding genomes. This coding genome is categorized as that is the less than 2% of the genome is referred to as the coding genome. These includes the genes that are basically transcribed and then translated into the specific proteins that have their structural, functional, and regulatory importance. And there are these specific portions of the genes that have correct instructions. So these are basically transcribed into the RNA and translated into the proteins. So the coding genome includes the genes and the exons. And when we talk about the non-coding genome, simply non-coding genome is further categorized into the three categories. These are referred to as the intragenic sequences, intergenic sequences, and the repetitive sequences. These intragenic sequences, these includes the introns. Introns are the non-coding regions within the genes. So these introns are basically not going to translate into the proteins, and these are the basically spliced out of the messenger RNA before the process of the translation. And the next are these UTRs. These are the untranslated regions that are not going to translate into the specific proteins. These are present outside of the start and stop codons. Next one are the intergenic regions. Intergenic regions are the specific non-coding regions that are present that are present between the genes that have no exactly known functions, but these are comes under the category of the regulatory functions. For example, promoters, enhancers, silencers. These promoters are basically required for the initiation of the transcription process. Next, these are the repetitive sequences. A major chunk of the sequences is the repetitive sequences. These are the lines, signs, etc. that we'll um, discuss in our next lecture. So if this helped you, then give it a like and please subscribe my channel for more videos. Thank you very much for watching.